Hello, and thank you for joining me on this gentle journey. Tonight, we drift high into the misty rainforest canopy, where the glass frog rests quietly beneath broad green leaves above flowing water. In this humid, softly lit world, rain moves in gentle patterns, and everything feels suspended in calm stillness. The glass frog's delicate, translucent body reveals the quiet rhythm of life within, as if nature itself has softened the boundary between what is seen and unseen. Let your mind settle into this peaceful canopy world where everything breathes slowly and nothing feels rushed. If you enjoy this relaxing moment, please consider liking, subscribing, or sharing your thoughts below. But for now, simply breathe and relax. Let's begin. Above the winding rivers of Central and South America, the rainforest canopy stretches like a living roof, suspended between earth and sky. It is a place where light is never still. Sunbeams fracture through overlapping leaves, scattering into shifting patterns of gold and green that drift with every movement of wind and branch. Beneath this upper world, water carves its ancient paths through stone and soil, carrying the constant memory of rainfall toward distant lowlands. The forest does not pause. It breathes in cycles of saturation and release, of storm and stillness, of humidity that clings to every surface. Within this layered environment, life organizes itself vertically as much as it does across the ground. Some creatures move through the leaf litter below, while others inhabit the branches, vines, and broad leaves that form a suspended ecosystem above the streams. It is here, in this elevated, humidity-rich corridor, that one of the rainforest's most delicate amphibians quietly exists. The glass frog does not dominate its surroundings. It merges with them, shaped by conditions that are neither fully aquatic nor fully terrestrial, but something in between. The leaves that extend over flowing water become critical structures in this world. They are not merely surfaces for photosynthesis, but platforms of survival. Rain gathers upon them and slides off in irregular rhythms, feeding the streams below and sustaining the constant dampness required by countless species. The air around these leaves is thick with moisture, so saturated that it clings to skin and membrane alike. In such conditions, amphibian life thrives in subtle ways, bound tightly to the presence of water without needing to remain submerged. The rainforest canopy above these streams is also a place of constant motion. Insects drift through the air in invisible currents, responding to temperature shifts and chemical signals carried by plants. Birds move through brief openings in the foliage, their presence marked more by sound than sight. Even the vegetation itself is not static. Leaves grow, twist, and reposition over time, slowly reshaping the architecture of light and shade. In this environment, stillness is rare, and survival depends on reading patterns that are constantly changing. For the glass frog, this world offers both opportunity and constraint. It occupies a narrow ecological band where humidity remains high enough to keep its skin functional and where access to water is always nearby. This proximity to streams is not accidental. It is essential. The frog's life is intertwined with the rhythm of rainfall and runoff, with the steady pulse of water moving through the forest's veins. Without this connection, its existence would quickly unravel. Yet, despite the abundance of life, the rainforest canopy is not chaotic. It is structured by invisible relationships between predator and prey, between moisture and leaf, between light and shadow. Every organism occupies a role shaped by millions of years of gradual adaptation. The glass frog fits into this system not as a dominant presence, but as a quiet participant in a far larger ecological conversation. As night approaches, the canopy undergoes a subtle transformation. The intensity of color fades, replaced by deeper tones of green and gray. Sounds become more pronounced, as if the forest is speaking in a lower register. It is within this shifting atmosphere that nocturnal life begins to stir, and the fragile balance of the ecosystem continues without interruption. The glass frog remains close to the underside of leaves, suspended above flowing water, part of a world that is at once stable in structure and endlessly dynamic in detail. Among the most striking adaptations in rainforest amphibians is the glass frog's translucent underside, a feature that allows internal structures to be visible through the skin when the animal is at rest. This characteristic is not uniform across the entire body, as the dorsal surface retains typical green pigmentation that matches surrounding foliage. The contrast between upper and lower surfaces creates a specialized form of concealment that functions in shaded vegetation environments above streams. In such settings, light is filtered through multiple layers of leaves, producing variable illumination that interacts with the frog's body in unusual ways. In anatomical terms, transparency in these frogs arises from reduced pigmentation in ventral tissues combined with structural properties of skin and muscle that permit greater light transmission. Unlike many vertebrates that rely heavily on pigments such as melanin for coloration and protection from radiation, glass frogs exhibit localized pigment suppression. This allows background light from leaves to pass through their underside, diminishing the visual outline when viewed from below. The effect is strongest when the frog is positioned against broad green surfaces, where transmitted light blends with surrounding wavelengths. Beneath the skin, internal organs such as the heart, liver, and portions of the digestive tract become partially visible. The heart's rhythmic movement can sometimes be observed through the abdominal region, especially during periods of reduced activity. Rather than being exposed in a vulnerable sense, these visible structures contribute to optical blending by breaking up continuous body outlines. The shifting shapes and tones within the body reduce sharp edges that predators might otherwise detect, particularly in environments where leaves cast fragmented shadows. Within the rainforest canopy environment, visual detection is often influenced by contrast rather than absolute color. The glass frog takes advantage of this by minimizing the contrast between its body and the leaf surfaces it occupies. When positioned against translucent or semi-translucent foliage, the frog's underside reduces silhouette clarity. This effect is enhanced by moisture on leaf surfaces, which further diffuses incoming light. As a result, the frog becomes difficult to distinguish from natural variations in leaf texture, especially in low-angle light conditions. From an optical standpoint, transparency in biological tissue depends on how light interacts with cells and fluids. In most animals, cellular structures scatter light, producing opacity. In glass frogs, reduced scattering in ventral tissues allows more direct transmission of light through the body. Water content in tissues also influences this property, as hydrated cells can align in ways that reduce refractive disruption. The combination of these factors produces an unusual balance between visibility and concealment that is highly effective in shaded arboreal habitats. Predatory species that hunt within the canopy often rely on motion detection, contrast sensitivity, and environmental cues beyond vision alone. Even so, reduced visibility offers a measurable advantage. When a glass frog remains motionless on the underside of a leaf, its outline becomes fragmented against the shifting background of veins, shadows, and moisture patterns. Birds and arboreal reptiles passing beneath may fail to register its presence unless movement occurs or environmental conditions change suddenly. Research into these amphibians has revealed that transparency in terrestrial vertebrates is relatively rare, making glass frogs a significant subject in studies of evolutionary adaptation. Observations suggest that their translucency is not an incidental trait, but a stable characteristic maintained through selective pressures in humid forest environments. The interaction between light diffusion, pigmentation distribution, and habitat structure continues to be examined to understand how such a form of concealment evolved and how it remains effective across different rainforest regions. Suspended above flowing rainforest streams, broad-leafed plants form elevated platforms that define the daily environment of glass frogs. These leaves, often belonging to shrubs and trees adapted to high humidity, extend horizontally or slightly downward over water channels, creating natural shelters that remain stable in a constantly shifting ecosystem. Their surfaces collect rainfall, channeling excess water toward edges where it drips into the stream below, maintaining a continuous connection between canopy and river. Positioning on the underside of these leaves is a critical behavioral trait. By occupying the lower surface rather than the upper, glass frogs reduce exposure to direct rainfall and many aerial predators. This orientation also provides a degree of concealment from organisms moving above the leaf layer. The underside tends to remain shaded, maintaining cooler and more stable microconditions compared to exposed upper surfaces that experience stronger evaporation and greater temperature variation. Humidity plays a central role in sustaining this habitat choice. Amphibian skin requires moisture for respiration and hydration balance, and the rainforest canopy offers consistent atmospheric saturation. Leaves overhanging streams benefit from constant evaporation rising from the water below, creating localized zones of high humidity that persist even during brief reductions in rainfall. Within these zones, glass frogs can maintain physiological stability without needing to travel long distances. Movement within this microhabitat is highly restricted. During daylight hours, individuals remain largely stationary, often adhering to leaf surfaces, using specialized toe pads that provide grip on smooth plant tissue. These adhesive structures allow them to withstand vibrations caused by wind, rainfall, or nearby animal activity. Remaining in one position reduces the likelihood of detection by predators that rely on motion to identify prey. As daylight fades, activity increases in subtle but important ways. Insect abundance rises within the canopy as nocturnal species become active, drawn to plant emissions and humidity gradients. Glass frogs respond by shifting short distances along leaf surfaces or nearby vegetation, positioning themselves along paths where prey movement is more likely. Their feeding range is typically limited to the immediate vicinity of their resting site, reinforcing the importance of leaf placement within the broader ecosystem. Leaf architecture itself influences survival outcomes. Species with broader, more stable leaves provide larger resting areas and better structural support for egg deposition. Leaves that hang closer to water surfaces reduce the distance hatchlings must fall to reach aquatic environments below. In contrast, smaller or more vertically oriented leaves may increase exposure or reduce reproductive success. This variability creates a patchwork of microhabitats, each with distinct advantages and limitations. Predator interactions are shaped heavily by leaf choice. Birds and arboreal reptiles moving through the canopy scan surfaces for irregularities in shape, color, or motion. When a glass frog remains still on the underside of a leaf, its outline blends with vein patterns and shadow gradients. Moisture droplets and natural imperfections in the leaf surface further disrupt visual continuity, making detection more difficult in filtered light conditions. Reproductive behavior is also tied directly to this environment. Many glass frog species deposit egg clusters on the underside of leaves above streams. This placement ensures that developing embryos remain moist while avoiding direct immersion in water, which could increase vulnerability to aquatic predators. The stability of the leaf surface is therefore essential, as it must support both adult frogs and developing eggs without detachment during rainfall or wind. Ecological relationships extend beyond the frog itself. Insects that inhabit or feed on these leaves contribute to nutrient cycling, while plant physiology influences humidity retention and surface texture. Each leaf functions as a small, self-contained habitat node within the broader rainforest structure, supporting multiple life stages of various organisms. Within this interconnected system, glass frogs occupy a specialized role defined by precision in placement and timing rather than range or dominance. When darkness settles across the rainforest canopy, the environment above the streams transforms into a layered field of faint movement and sound. In this low-light setting, glass frogs shift from near-still positioning on leaf undersides to carefully timed feeding activity. Their behavior is closely tied to nocturnal insect abundance, which rises as moths, flies, ants, and small arthropods become active within humid air, currents drifting between vegetation. Vision plays a central role in this transition. Large, forward-facing eyes, adapted for low-light conditions, allow glass frogs to detect subtle motion against the dim background of leaves and water reflections. These visual systems are sensitive to contrast changes rather than fine detail, enabling detection of small prey moving across irregular surfaces. The rainforest at night provides limited illumination, but reflected light from water and scattered moonlight filtering through the canopy creates enough visual structure for hunting. Movement during feeding is highly economical. Instead of pursuing prey across long distances, glass frogs rely on stationary positioning and rapid strike behavior. When an insect enters a reachable range, the frog extends its tongue in a swift motion that captures the target before it can escape. This method reduces energy expenditure and minimizes exposure time in open positions where predators might detect movement. Air currents above the streams influence prey distribution in significant ways. Warm, moisture-rich air rising from water surfaces carries insects upward toward leaf layers, where they often land briefly before continuing flight. These short resting moments create predictable opportunities for capture. Glass frogs position themselves along leaf edges or undersides, where insect traffic is statistically more frequent, increasing the likelihood of successful feeding events. Auditory and vibrational cues also contribute to hunting efficiency. While vision remains primary, Subtle disturbances in leaf surfaces caused by insect landings can transmit mechanical signals through plant tissue. Toe pads on the frog's feet detect these vibrations, providing additional information about nearby prey presence. This multisensory integration allows for accurate targeting even when visual clarity is reduced by shadow or fog. Prey selection is generally opportunistic, focusing on small insects that can be subdued quickly. Flies and ants constitute a significant portion of the diet due to their abundance in humid environments. Moths and other nocturnal insects become more prominent during periods of increased floral activity or changes in atmospheric pressure that influence insect flight patterns. Feeding frequency varies depending on environmental conditions and local insect density. Energy balance is a critical factor in this feeding strategy. Glass frogs maintain relatively low metabolic rates compared to many active predators, allowing them to survive on intermittent feeding rather than continuous intake. This metabolic adaptation aligns with their sit and wait approach, where long periods of stillness are followed by brief bursts of activity. Such a strategy reduces exposure to predation while maintaining sufficient energy reserves. Predator avoidance remains an underlying constraint during feeding periods. Nocturnal hunters, such as certain snakes and birds of prey, may also move through canopy layers, relying on motion detection or heat sensing rather than vision alone. To reduce risk, glass frogs often limit movement to minimal strike actions and return quickly to stationary positions after feeding attempts. This balance between energy acquisition and safety shapes nearly every aspect of their nocturnal behavior. Environmental variability influences feeding success across different nights. Rainfall intensity, wind movement, and insect population fluctuations all alter prey availability. During heavy rainfall, insect flight may decrease, reducing feeding opportunities, while mild, humid conditions often increase activity levels. Glass frogs respond to these shifts through flexible timing, adjusting activity intensity according to immediate conditions rather than fixed schedules. Within this nocturnal feeding landscape, survival depends on precision, timing, and restraint. Each strike represents a brief intersection between predator and prey, embedded within a broader ecosystem where countless interactions occur simultaneously above the flowing waters of the rainforest. Above the steady flow of rainforest streams, reproduction in glass frogs unfolds on the underside of leaves that extend over moving water. This placement is a defining feature of their life cycle, linking terrestrial surfaces directly to aquatic environments below. Egg deposition occurs in clusters, with gelatinous masses attached to leaf tissue using adhesive secretions that secure them against rainfall, wind, and minor mechanical disturbances. Humidity plays a crucial role in the survival of these developing embryos. The rainforest canopy provides consistently moist air, preventing desiccation while allowing gas exchange through the egg membrane. Without this atmospheric saturation, embryonic development would be severely compromised. Leaves positioned above streams are especially valuable because evaporation from the water below helps maintain stable microclimatic conditions around the egg masses. Parental behavior varies among species, but in several glass frog lineages, males remain close to egg clutches after deposition. This guarding behavior involves positioning near or directly on the egg masses, where the adult can deter small predators such as ants, parasitic insects, and certain mites. The presence of a guarding male reduces egg mortality, particularly during early developmental stages, when embryos are most vulnerable. Development within the eggs follows a gradual sequence of embryogenesis, during which visible changes occur as cells differentiate into functional organ systems. Over time, embryos begin to exhibit movement inside the gelatinous matrix, signaling progression toward hatching readiness. This stage is highly sensitive to environmental stability, with fluctuations in humidity or temperature affecting developmental timing and survival rates. Hatching represents a critical transition between terrestrial and aquatic life. When development reaches completion, tadpoles emerge from the egg capsule and drop directly into the stream below. This descent is not random, but synchronized with their readiness for aquatic adaptation. Upon entering the water, the larvae immediately begin a new phase of life, relying on stream currents and submerged vegetation for shelter and food sources. Aquatic development introduces a different set of ecological pressures. Tadpoles must navigate flowing water, avoid aquatic predators, and locate suitable feeding areas. Their morphology is adapted for this environment, with tail structures that facilitate swimming and mouthparts designed for grazing on algae and organic matter. Streams beneath rainforest canopies provide both nutrients and shelter, but also contain diverse predator populations that influence survival rates. The positioning of egg deposition above water reduces certain risks associated with aquatic egg laying, such as direct predation by fish or aquatic invertebrates. However, it introduces alternative threats, including exposure to aerial predators and environmental instability caused by wind or heavy rainfall. The balance between these risks reflects a long evolutionary history of adaptation to arboreal reproductive strategies. Leaf stability becomes a determining factor in reproductive success. Leaves must remain intact and attached to their supporting branches throughout the incubation period. Structural failure caused by storms or natural decay can result in egg loss, making plant selection an important aspect of reproductive behavior. Females often choose leaves with sufficient surface area and mechanical strength to support both egg mass weight and environmental stress. Interactions between adult frogs and developing embryos highlight a tightly linked life cycle that spans multiple habitats. The connection between canopy leaves and flowing streams ensures that offspring transitions seamlessly from aerial deposition sites to aquatic environments. This dual dependence on terrestrial and aquatic systems defines the reproductive strategy of glass frogs and reinforces their reliance on intact rainforest ecosystems. Within this reproductive framework, survival is shaped by timing, placement, and environmental stability. Each stage, from egg deposition to larval emergence, is embedded within a broader ecological network that connects rainfall, vegetation structure, and stream dynamics into a continuous system supporting amphibian life. Across the biological world, transparency is rare in land-dwelling vertebrates. Yet glass frogs exhibit a striking version of it along their ventral surfaces. This characteristic is not a simple absence of color, but a structured optical condition produced by cellular organization, pigment distribution, and tissue composition that together reduce light scattering within the body. At the microscopic level, most animal tissues appear opaque because light is deflected by cellular boundaries, organelles, and pigment granules. In glass frogs, the ventral skin contains significantly reduced pigmentation, which lowers absorption of light in specific wavelengths. More importantly, Internal structures are arranged in a way that minimizes disruption of light paths passing through the body when it is pressed against translucent leaf surfaces. Within rainforest environments, this optical adaptation becomes especially effective due to the nature of ambient lighting. Sunlight filtering through multiple canopy layers reaches lower vegetation in fragmented patterns, producing overlapping zones of brightness and shadow. Against this background, transparency does not function as complete invisibility, but rather as a reduction of visual contrast, making detection by predators more difficult. A key factor in this phenomenon is the interaction between skin hydration and optical clarity. Amphibian skin is highly permeable, allowing for continuous exchange of water and gases. In glass frogs, this permeability supports a stable level of tissue hydration that influences how light passes through cells. Well-hydrated tissues tend to exhibit reduced refractive variation, which decreases scattering and enhances translucency. Internal organs contribute to the complexity of this optical system. The heart, liver, and digestive structures remain functionally active while partially visible through the ventral body wall. Rather than creating a clear silhouette, these organs introduce internal visual complexity that disrupts the formation of a consistent outline. In environments where predators rely on edge detection and contrast recognition, this internal variation reduces the likelihood of accurate targeting. Leaf surfaces play an essential role in reinforcing this adaptation. When a glass frog rests on the underside of a leaf, the green pigmentation and vein networks create a layered visual field. Light passing through the frog's body interacts with this background, blending structural outlines with natural patterns. Moisture on leaf surfaces further diffuses incoming light, softening edges and reducing sharp transitions between body and environment. Evolutionary biology suggests that such transparency likely developed under selective pressure from visually oriented predators in arboreal habitats. Birds and reptiles that forage within the canopy often depend on detecting subtle differences in shape and shading. Any reduction in contrast provides a measurable survival advantage, even if it does not eliminate visibility entirely. Over time, individuals with greater translucency would have experienced improved survival rates in specific microhabitats, reinforcing the trait across generations. Comparative studies of amphibians indicate that full-body transparency is uncommon in terrestrial species due to structural limitations and exposure risks. Glass frogs represent a specialized case in which partial transparency is sufficient to enhance concealment without compromising physiological function. This balance between visibility and biological integrity is a defining feature of their anatomy. Environmental conditions also influence the effectiveness of this adaptation. Humidity levels, lighting angles, and leaf texture all affect how light interacts with the frog's body. Under high moisture conditions, optical blending becomes more pronounced, while drier or harsher lighting can reduce its effectiveness. This dependence on external variables highlights the close relationship between physiology and habitat structure. Research into these mechanisms continues to reveal how subtle modifications in tissue composition can produce significant ecological advantages. The glass frog demonstrates that transparency in vertebrates is not an all-or-nothing trait, but a graded condition shaped by multiple interacting biological and environmental factors within the rainforest canopy system. Beneath the thin ventral surface of a glass frog, internal organs are not fully concealed, but instead partially exposed through a layer of translucent tissue. Among the most visually distinct of these structures is the heart, which can often be observed as it contracts rhythmically within the thoracic cavity. This visibility is not a structural weakness in evolutionary terms, but a byproduct of reduced pigmentation and specialized tissue organization that allows light to pass through the body with limited scattering. Within rainforest canopy conditions, this anatomical transparency interacts directly with environmental background patterns. When the frog rests against a leaf underside, the movement of internal organs blends into shifting textures of green pigmentation, vein networks, and moisture gradients. The heartbeat, although externally observable in controlled conditions, becomes significantly harder to isolate visually when combined with these complex background elements. Physiological function remains fully intact despite external visibility. The cardiovascular system operates in a standard amphibian configuration, circulating oxygenated blood through a closed loop that supports both muscular activity and skin-based respiration. Amphibians rely heavily on cutaneous gas exchange, and glass frogs are no exception. Their skin permits the diffusion of oxygen and carbon dioxide, supplementing pulmonary respiration and reinforcing the importance of maintaining constant hydration in humid environments. At the cellular level, reduced pigmentation contributes to optical clarity, but does not eliminate structural integrity. Melanocytes, which produce melanin in many vertebrates, are less concentrated in ventral regions. This reduction decreases light absorption and helps maintain translucency. However, dorsal surfaces retain typical green pigmentation, which provides camouflage when viewed from above, demonstrating a dual-layered strategy of concealment depending on viewing angle. The beating heart, while visible through the skin, does not create a clear outline in natural conditions. Instead, its motion is fragmented by overlapping tissue layers and the curvature of the body. In environments where light is diffused through foliage, this movement becomes part of a broader pattern of shifting brightness rather than a distinct signal. Predators relying solely on vision may struggle to distinguish such internal motion from environmental variation. Blood circulation in glass frogs follows typical amphibian physiology, but oxygen transport is closely linked to environmental moisture levels. Because amphibian skin participates in gas exchange, hydration directly influences respiratory efficiency. In humid rainforest zones, this system operates effectively, allowing internal organs to function without reliance on constant lung ventilation. This physiological flexibility supports survival in variable microclimates within the canopy. Microscopic examination of transparent tissue reveals a tightly organized cellular structure that reduces refractive discontinuities. Cells are arranged in a way that minimizes abrupt changes in light direction as it passes through the body. This arrangement does not eliminate scattering entirely, but it reduces it enough to produce partial translucency. The result is a body that reveals internal anatomy without becoming fully exposed in visual terms. Predation pressure continues to shape the significance of this trait. Many canopy predators detect prey through movement patterns, contrast differences, or heat signatures, rather than relying exclusively on shape recognition. In this context, visible internal organs do not necessarily increase detection risk as long as external outlines remain disrupted and environmental blending remains effective. During periods of rest, when muscular activity is reduced, internal visibility becomes more pronounced. The heart's rhythmic contractions and occasional digestive movements may be observable through the ventral surface. Even so, these signals are often obscured by shifting leaf textures and changing light angles caused by wind movement in the canopy above. Comparative amphibian studies indicate that such partial transparency is unusual among vertebrates inhabiting terrestrial environments. Most species rely on opaque tissues for protection against radiation and structural reinforcement. Glass frogs, however, represent a specialized adaptation where selective translucency coexists with fully functional organ systems, shaped by the unique demands of humid arboreal ecosystems. Within this biological framework, visibility and concealment are not opposing forces, but interdependent features of a single adaptive strategy. Internal life remains active and observable under certain conditions, yet continuously integrated into a broader environmental context that determines whether it is perceived or overlooked. Within the layered structure of tropical rainforests, survival for glass frogs is closely tied to localized atmospheric conditions that exist on scales far smaller than the broader ecosystem. These microclimates are formed by the interaction of dense vegetation, continuous water cycling, and persistent evaporation from streams running beneath the canopy. In such environments, humidity is not a background feature, but a defining physical condition that shapes amphibian physiology and distribution. Air surrounding leaf surfaces above streams often reaches near saturation levels of moisture. This saturation results from constant evaporation rising from moving water below, combined with transpiration from surrounding plants. As water vapor accumulates within these enclosed spaces, it creates stable zones where amphibian skin can maintain proper hydration. For glass frogs, this stability is essential, since their permeable skin allows continuous exchange of gases and water with the environment. Leaf positioning relative to water sources plays a central role in maintaining these conditions. Vegetation that extends over flowing streams benefits from a consistent upward flux of moisture, which prevents rapid drying even during brief reductions in rainfall. These conditions form narrow ecological bands where temperature variation is reduced and humidity remains relatively constant throughout daily cycles. Within these bands, glass frogs find the environmental stability required for both survival and reproduction. Physiological dependence on external moisture means that even minor environmental shifts can influence behavior. When humidity drops, amphibians may reduce activity or relocate to more sheltered leaf surfaces closer to water flow. Conversely, periods of heavy rainfall increase atmospheric moisture and expand suitable habitat areas temporarily, allowing broader distribution across canopy layers. Rainfall patterns in tropical regions contribute directly to the formation and maintenance of these microhabitats. Water intercepted by leaves drips in irregular intervals, while excess runoff channels towards streams below. This continuous movement of water ensures that evaporation and condensation remain in balance, reinforcing the humid conditions required by moisture-dependent organisms. Without this cycle, ventral skin surfaces would rapidly lose hydration, impairing essential respiratory functions. Gas exchange through amphibian skin is highly sensitive to environmental moisture levels. In glass frogs, cutaneous respiration supplements lung function, allowing oxygen and carbon dioxide exchange across the skin surface. This mechanism becomes less efficient in drier conditions, reinforcing the species' restriction to humid forest zones. The surrounding microclimate therefore acts as an external regulator of physiological stability. Temperature variation within these environments is moderated by canopy density. Thick layers of vegetation absorb and diffuse incoming solar radiation, preventing rapid heating of lower surfaces. As a result, thermal fluctuations remain relatively narrow compared to open environments. This stability supports metabolic regulation in small ectothermic organisms that depend on external heat sources to maintain activity levels. Hydrological connectivity between canopy and stream systems reinforces the ecological dependence of glass frogs on intact forest structure. Water cycling through evaporation, condensation, and precipitation links aerial and aquatic environments into a continuous system. Any disruption to this cycle, such as deforestation or changes in rainfall patterns, can alter humidity distribution and reduce suitable habitat availability. Ecological sensitivity to these conditions makes glass frogs useful indicators of environmental change. Shifts in their presence or behavior can reflect broader alterations in humidity regimes, vegetation density, or water availability. Because they occupy such narrow environmental niches, even small disruptions can produce noticeable effects on local populations. Within this framework, survival is determined by the interaction between physiology and atmospheric stability. The rainforest does not merely surround glass frogs. It regulates their internal function through moisture, temperature balance, and water flow. Each leaf, stream, and vapor cycle contributes to a finely structured system in which amphibian life persists only where conditions remain within a narrow and carefully maintained range. Across the rainforest canopy, survival for glass frogs is shaped by a constant presence of predation pressure from multiple directions and species. Birds moving through foliage, arboreal snakes navigating branches, and a range of invertebrate hunters all contribute to an environment where detection can lead quickly to capture. Even with specialized camouflage, these amphibians exist within a complex web of biological threats that operate continuously across day and night cycles. Visual concealment provides only partial protection. Transparency reduces contrast against leaf surfaces, but many predators rely on additional sensory systems beyond sight. Snakes, for example, can detect chemical cues and vibrations transmitted through vegetation. Certain birds use rapid scanning combined with motion sensitivity to identify small organisms resting on leaves. Invertebrate predators, including large arthropods, may respond to proximity and tactile cues rather than visual outlines alone. Stillness during daylight hours is a primary survival strategy. When light filters through the canopy and creates shifting patterns across leaf undersides, any movement increases the likelihood of detection. Glass frogs therefore remain in fixed positions for extended periods, minimizing muscular activity and reducing environmental disturbance around their bodies. Even minor shifts in posture are limited, as subtle motion can alter the leaf's surface tension or create detectable vibrations. At night, activity increases as feeding becomes necessary, but this period introduces its own risks. Darkness does not eliminate predators. Instead, it changes the types of threats present. Nocturnal snakes and amphibians move through the canopy, while certain birds may still hunt under low-light conditions. Glass frogs must balance the need for energy intake with the requirement to remain inconspicuous during movement. Energy constraints further shape behavioral patterns. As small ectothermic organisms, glass frogs depend on external environmental conditions to regulate metabolism. Their energy reserves are limited, requiring efficient feeding strategies that reduce unnecessary movement. This constraint reinforces sit-and-wait hunting behavior, where short bursts of action replace prolonged activity. Chemical signaling in the rainforest adds another layer of complexity. Many organisms release pheromones or metabolic byproducts that can be detected by specialized sensory organs in predators. Amphibians with permeable skin are particularly susceptible to chemical detection, which means that humidity and airflow patterns can influence exposure risk. Wind currents moving through vegetation may carry scent molecules beyond immediate surroundings, increasing vulnerability even when visual detection is minimized. Predation pressure also varies with spatial positioning within the canopy. Leaves closer to stream edges may offer increased humidity, but also greater exposure to animals moving between water and vegetation. Higher canopy levels can reduce some ground-based threats, but introduce increased exposure to avian predators. Glass frogs must therefore occupy positions that balance these competing risks, often selecting leaves that provide both concealment and proximity to water. Structural features of vegetation contribute to survival outcomes. Leaves with complex vein patterns, irregular edges, or overlapping surfaces provide better visual disruption compared to smooth or sparse foliage. Moisture accumulation on leaf surfaces can also alter light diffusion, further reducing visibility. These environmental variables interact continuously with frog behavior, shaping microhabitat selection and resting orientation. Predation risk is not uniform across time. Seasonal changes in rainfall influence predator abundance and activity levels, while fluctuations in insect populations indirectly affect feeding opportunities and exposure duration. During periods of high biological activity, interactions between species increase, raising the likelihood of encounters across trophic levels. Within this ecological context, glass frogs occupy a position defined by vulnerability and adaptation. Their survival depends on reducing detection probability across multiple sensory channels, while maintaining enough activity to sustain metabolic needs. The balance between concealment and necessary movement defines nearly every aspect of their behavior within the rainforest canopy system. Within tropical ecosystems, certain species serve as sensitive indicators of environmental stability, and glass frogs occupy a particularly responsive position in this regard. Their dependence on consistent humidity, intact vegetation, and clean freshwater systems makes them highly sensitive to even minor ecological changes. Because of this sensitivity, fluctuations in their populations often reflect broader environmental shifts occurring within rainforest habitats long before those changes become obvious in larger, more resilient species. Forest structure plays a foundational role in maintaining suitable conditions for these amphibians. Dense canopy cover regulates humidity by limiting direct solar exposure and reducing evaporation rates at ground and mid-canopy levels. When vegetation is fragmented through deforestation or selective logging, these regulatory effects weaken. As a result, localized microclimates become more variable, with reduced moisture retention and increased exposure to wind and sunlight. Water quality in stream systems beneath the canopy is equally important. Glass frogs rely on clean, oxygen-rich streams for the aquatic stage of their life cycle, where tadpoles develop after hatching from leaf-based egg deposits. Sedimentation, chemical runoff, or changes in water flow can alter habitat conditions for larvae, reducing survival rates and disrupting population stability. Because these streams are directly connected to surrounding vegetation, changes in forest structure often translate quickly into aquatic impacts. Climate variation adds another layer of complexity. Shifts in rainfall patterns influence humidity levels across the canopy, affecting both adult physiology and reproductive success. Extended dry periods reduce available moisture on leaf surfaces, making egg clutches more vulnerable to desiccation. Conversely, intense rainfall events can physically dislodge eggs or alter stream dynamics, increasing mortality risk during early developmental stages. Human activity introduces additional pressures on these interconnected systems. Habitat fragmentation reduces continuous forest cover, isolating populations and limiting genetic exchange between groups. This isolation can reduce resilience over time, making populations more vulnerable to disease, environmental fluctuation, and stochastic events. Even small breaks in forest continuity can significantly alter movement patterns and breeding site availability. Pollution represents another significant factor influencing amphibian health. Amphibians, due to their permeable skin, are particularly susceptible to chemical changes in their environment. Contaminants entering waterways can interfere with larval development or disrupt physiological function in adults. Because glass frogs rely on both terrestrial and aquatic environments, exposure can occur at multiple life stages. Even low concentrations of pollutants may accumulate across development, affecting growth rates, survival probability, and reproductive success in sensitive rainforest populations. Ecological monitoring has shown that amphibian declines often precede visible changes in other animal populations. This early response is linked to their dual dependence on land and water systems, which amplifies their sensitivity to environmental disruption. In this context, glass frogs function as biological indicators, offering insight into the stability of rainforest ecosystems through changes in their abundance and distribution. Because they rely on both canopy microclimates and clean freshwater streams, they respond quickly to shifts in humidity, pollution levels, and habitat fragmentation. These responses allow researchers to detect ecological stress at an early stage, providing valuable data for understanding long-term environmental trends and guiding conservation priorities. Conservation strategies aimed at protecting these amphibians typically focus on preserving continuous forest corridors and maintaining stream integrity. Protecting canopy structure ensures that humidity levels remain stable, while safeguarding waterways supports successful larval development. These measures also benefit a wide range of coexisting species that rely on similar environmental conditions. In practice, this often involves limiting deforestation, reducing fragmentation from roads and agriculture, and preserving riparian vegetation along stream banks. Restoration efforts may include reforestation with native plant species to rebuild canopy cover and reconnect isolated habitats. By maintaining ecological continuity between trees and freshwater systems, conservation work helps sustain the microclimates essential for amphibian survival and long-term population stability. Research into glass frog populations continues to contribute to broader ecological understanding of tropical rainforest dynamics. Their presence, absence, or behavioral changes provide measurable signals about ecosystem health, linking small-scale biological responses to large-scale environmental trends. Through this relationship, they offer a biological perspective on how interconnected rainforest systems respond to disturbance over time. As sensitive amphibians dependent on stable humidity, intact vegetation, and clean stream networks, even minor environmental shifts can be reflected in their distribution and reproductive success. Continued monitoring of these patterns helps researchers identify early indicators of ecological stress and better understand how rainforest environments adjust to ongoing climatic and structural change. Within this framework, glass frogs represent more than a single amphibian lineage. They function as part of a larger environmental feedback system, where changes in vegetation, water, and climate are reflected in the structure and stability of living populations across the canopy and stream network. Subtle shifts in humidity levels, canopy density, or stream clarity can alter breeding success, movement patterns, and long-term population distribution. Because their life cycle is tightly bound to both terrestrial leaves and aquatic streams, even localized disturbances propagate through their biology. Over time, these responses form measurable signals that reveal the condition of the surrounding rainforest, linking individual survival to ecosystem-wide balance. As our journey through the rainforest canopy comes to a close, picture the glass frog resting quietly on a leaf above flowing water, its world fading into soft greens and distant rain. The forest grows quieter, wrapped in mist and gentle sound. There is something deeply calming in its transparency. Nothing hidden, nothing forced, only quiet existence in harmony with its surroundings. Let the gentle sensations of peace and calmness settle through your body and mind. Feel your thoughts slow, your breathing deepen, and your body soften into rest. If you enjoyed this relaxing moment, please consider liking, subscribing, or sharing your thoughts below. You can also turn on the notification so you don't miss any of our future journeys together. But for now, let your body relax, release all tension, and allow your mind to drift gently into rest. Good night and sleep well.